The next speaker is Dr. Vamsi. Dr. Vamsi is a PhD from Penn State in theoretical computer science, and his present focus includes text mining, data visualization, and NGS data analysis. The topic of his talk today would be knowledge representation and analysis. And I'd like to hand over to Dr. Vamsi.
Hi everyone. So this is not an area of which I have very deep knowledge, but there's a lot of work on this area recently because you want to figure out how to interpret the genome. So far what we discussed in Shanmukh's talk and what Chandra had mentioned is that we know how to read the genome, we know how to sequence it, we know how to find all the variants and all that. But if you sequence a typical human genome, you will get around 4.5 million variants. So if you take any one of your genomes and do all of the sequencing, you compare it against that human genome sequence that's there at NCBI, out of those 3 billion locations, you'll find around 4.5 million locations which are different. So you have all of this data, but you don't know how to interpret them. You don't know which of those variants are important, which of them are not. So even if you were to look only within the genes, so in the entire 3 billion genome, around 10% of it or 5% actually is the genes, and the rest of it is what nobody really knows what it does. So even if you were to look only within the 30,000 genes, you'll get around 50,000 variants. So then if you take somebody who has a disease and he goes to the doctor and he thinks, it most probably is a genetic cause, and you sequence that person and you find this 50,000 variants, you don't know which of those genes is important, which of those 50,000 variants is actually responsible for the disease. So for that you need to go back to the data and all of the wisdom that has been accumulating in biology and say, how do we get a handle on all of that knowledge? How do we store that knowledge? How do we query that knowledge, and so on? So I'll be talking from that perspective. Okay. So most of what we do in Strand is of a very quantitative nature, which mainly involves measurements. So you're trying to measure how active is a gene. So it's something called expression of a gene. So you can take a particular cell in a particular tissue and say how much expression is every gene showing. So you have 30,000 genes, so you might have 30,000 measurements. Then you could look at it in a different part of the body and say, for instance, you ask what makes a blood cell different from some other cell. That's because the genes that are expressed in blood cells will be very different from the genes that are expressed in liver cells. Okay. Similarly, it's not just the genes become messenger RNA. The messenger RNA is translated into proteins. So you have around 100,000 distinct proteins in the human body. So the protein levels will also be different from one cell to the other cell. And then you have metabolites. Metabolites are not genes or proteins. There, if you eat some food like we had right now, you got the glucose and the glucose got converted into something else. Those are all chemical molecules. So you can measure metabolite concentrations. So a lot of biology is about how do you measure all of these things accurately and in a high throughput fashion. So for instance, if you were to, in a very classical setting, you're trying to find out the cause for one particular disease. So you believe that there might be some gene which is behaving differently between the healthy people and the diseased people. So how do you go about it, right? So you take the measurements of all the genes because you have no idea which genes to focus on. So you take a bunch of healthy people, you take a bunch of people with the disease, you measure all of their genes, and then you start looking for patterns. You want to find out, is there one gene or a collection of genes out here whose values are very different between the diseased population and the healthy population. So these are all classical quantitative stuff, and we've been doing tools for this. So the complications arise from the fact that you can't just have two populations of people saying diseased and healthy. Within the diseased population, you'll have them from different ethnicities, you'll have different ages, you'll have different genders. Some of them will be smokers, some of them will be non-smokers. So you want to make sure that whatever genes that you're finally narrowing down on, saying these genes are behaving differently, you want to make sure the difference is because of the disease and not because of any of those other parameters. So you have this huge multidimensional space where you have people, they have an age, they have a gender, they have ethnicity, they have disease, they have weight, smokers, non-smokers, all of that stuff. And they have protein measurements or gene measurements associated with them. And you want to find out, given all of these parameters, are there any genes which are behaving differently? And once you find out some statistically significant genes, then you start trying to figure out why are these genes behaving differently, what is the mechanism, how could that particular gene actually be involved in the disease, and so on. So all of this is traditional bioinformatics work, toxicogenomics work, all of them are based on this paradigm. Most of you are mathematicians, statisticians, so all of these problems might sound familiar to you. But I'm going to focus on a slightly different topic, more on the qualitative side of things. So we've been talking about all of these genes, but how do we describe the gene's function? So we have these 30,000 genes. We wanted to take a gene and describe what it does. How do you describe it? What is the vocabulary for it? And a lot of the knowledge that we have about a gene comes from our knowledge of the same gene in other organisms. So you might look for the equivalent gene in the mouse. You might do a lot of mouse experiments and describe what the gene does in the mouse. Or maybe you could not do those experiments in the mouse, you do it in the zebrafish or Drosophila or yeast and so on. So what is the standard vocabulary in which you will describe a gene's function? Similarly for a disease, how do you describe disease? If a patient walks in and the patient has a lot of symptoms, how do you describe those symptoms? And it's very important to have a standardized way of describing all of these so that you can reason on top of it.
These genes are defined in terms of their expressions only?
So there's a nomenclature committee which meets a couple of years, and some time back they said, you know what, we should outlaw the word gene because it's become so ambiguous, nobody really knows what to call a gene. But finally it is still accepted that anything which is expressed and might have a functional role is a gene. It's as vague as that.
So that collection,
that portion of the genome. Yes. So initially 30,000 such loci. Yeah. So initially when the human genome was sequenced, the bet was that they had a drop box and people would vote on, bet on how many genes they would find in the human genome once it was actually done. And they thought that because the lower organisms like C. elegans and the worm and all have around 5,000 genes, and they thought we are so much more advanced, we should have around 100,000 genes, maybe 1 million genes and so on. Finally the number has been dwindling every year, now it's around 25,000 genes.
and then a huge number you don't know how to.
There's a huge portion of the genome that has some controlling circuitry, but you don't really, you haven't really deciphered that yet.
To map, let us say mathematically, if you map a gene, whatever that means, to a set of physiological function, is that map, I'm sure it's not one-to-one.
Yeah, it's not.
It's many-to-many, or what would you say?
Yeah, it is many-to-many. There are some core set of genes which have, so genes occur in something called gene families, and different members of the gene family usually have similar functions. And in different organisms, these gene families are of different sizes. For instance, if you take all of our smelling related genes, olfactory genes for instance, in human you have a small set of them, in mice you have around 600 genes in that family, because they have more of a need for it. I mean, you could say have more of a need. It's not like they had a need and so the genome duplicated, but it was sort of those rodent families which had a lot more of those genes and which had a lot more sensitivity there flourished, and the other rodent species might have died out, things like that. So each organism, we differ a lot from the chimps and all in our immune genes. A lot of the other genes are almost exactly the same. The immune genes are very, very different.
Do we know anything which characterizes, which genes determine the brain? Or
No, it's there are two ways of answering that. One is they have done, they have taken the different tissues and studied in each tissue which genes are expressed. So they have an idea of what all are the set of genes which are expressed more in the brain than in the other tissues. So you know that those genes have something to do with the brain, but exactly saying, draw me a flow chart saying how do these genes in the brain affect, result in the brain evolution, that part it's very hard to do.
And the expression is only in terms of proteins, is it?
Yes.
Unique, so it is one-to-one.
Not really. A gene has something called multiple isoforms. So each gene will typically give rise to 2 different types of RNA on an average, and those RNA get translated into proteins, and the proteins form complexes afterwards, and the proteins undergo modifications. So you might have 25,000 genes, you might have around 50,000 mRNA, different types of mRNA, and you'll have around 100,000 proteins. Okay.
So that is the map.
Yeah.
so just one last question. So if mathematically I want to represent the expression let us say, or the manifestation of a gene, as let us say you can treat it as a vector in 3 billion dimensional space, whatever that means. There is a set of all the base pairs to an individual in the human and so on and so forth. Now what you are saying is basically you can you can actually partition this vector into gene classes and further into genes, and essentially each gene class would affect one dimension. I don't know whether we have come to this this level of understanding. Can we identify for each gene class what physiological dimension is determined? Again, we'll have rough knowledge in several cases.
I think this issue is probably very complex.
Yeah.
Because how does a gene get switched on? How does it get switched on?
Yeah, so
Orchestrates that and at what level?
yes.
I don't know. What kind of feedback mechanisms exist and
Yes.
for this to happen?
So there are some NGS experiments which people do purely to determine the controlling circuitry for a gene, and vice versa also. There are around 2,500 genes called transcription factors which really control the expression of other genes. So those are like master circuit. There's feedback circuits. Yeah.
That's what complicates enormously everything.
Yeah. So for instance, there are some things which have to respond very rapidly. There's a class of proteins called heat shock factors. So if the temperature goes up a little bit, you want those heat shock factors to be immediately start generating proteins and carrying out certain activities. So, but then when the temperature comes back to normal, you want them to go down on their activities. So it's very, very fast acting feedback loops and very complicated circuitry out there. But to understand just for each gene to understand its circuitry, it's almost like an entire lab's work over 20 years it takes.
Do they understand if I isolate one gene and one physiological dimension, do we have a simplified dynamical system formulation for it? I'm sure for some examples there is.
Yeah.
But what is sort of, if somebody asked a general question, do we know the dynamics of expression? And
yeah, there are some labs which focus on it, but it's not just one transcription factor. Now they're finding out there's regions called promoters, there are transcription factors, there are enhancers, there are suppressors. So the huge army of proteins and their concentrations together which determine whether a gene is switched on or not. So to model all of that, other than specific examples, nobody has a master theorem for that yet.
And typically, if I just one last. If I once looked at some cell model, and actually I looked at your liver model also very briefly, an early version of it, 100 parameters and everything's non-linear. So Veerachaneni did shed some light on this. But do we have, let us say, the beginnings of a mathematical theory of such even?
I don't think any such broad unified theories will emerge, but I think if you have a specific endpoint in mind, it might be possible to model it. So they've taken maybe the glucose system and said, can we identify all of the actors in the glucose system and what are the feedback loops? For such small systems you can do it, but not at an entire organism level. Okay. So Craig Venter, that's what Craig Venter is trying to do by creating the synthetic cell, because the cell should be able to divide on its own, it should be able to create its own content, it should be able to replicate on its own completely from scratch. Now the challenge is, if you start with something as complex as the human cell, which has 30,000 genes and all of this controlling circuitry you don't know about, it's too hard to start off. So they had a lot of work on what is the minimal organism we can create, what all genes can we keep dropping off, and choose a smallest set of maybe 400 genes or so and try to create a viable organism. And after they created that viable genome, it still wasn't, it didn't have the bootstrap capability. It couldn't start off from an inorganic state itself. So then they injected that into an organic existing cell. But once this thing was switched on, it sort of replaced the host, proving that you need some starting matter, but once you use that to bootstrap it, it kind of creates its own self. So
has it been that particular one? How many generations have so far?
I think they've shown that in a tissue culture, in a lab facility, it can continue to replicate itself. But I haven't kept up to date on that.
But we don't have anywhere near a theory, mathematical theory, or
There are small theories for individual parts, but not a big overall theory.
Do we have at least a mathematical theory for which can explain the folding of a prion?
Individual.
From okay, I know that you will say no from the Schrodinger wave equation. Almost certainly the answer is no, but I have not tracked the literature.
Yeah, structural bioinformatics I don't know much about.
No, I said Schrodinger equation.
Yeah, I mean there is that minimum energy state that you want to do. I mean if protein, if you model all of the force fields for it and say it will fold into the minimum energy configuration, that is what most of the softwares are based on. It's just that finding that minimum energy configuration is so time consuming that you need supercomputers. People start off with homology models, they start with some structure which is equally known.
Energy, when you want to minimize free energy,
Yeah.
the complication is because of the water molecule.
Yeah.
And all the entropy related computations, it gets very complicated. I think there is a competition that's held every 2 years for folding.
Yeah.
And that the CAST is called it. And last few years it's been won by
D. Shaw, Raval's group.
He has built actually a high performance machine that is actually tailored for this type of computation. Out of all the money he made from the hedge fund, he's put some into this. He's gone back and become a professor again at Columbia. This has been
Yeah, but they're having trouble coming up with the exact force fields. I mean,
right.
Those are all heuristic, right?
Yeah,
I mean, okay. So here, in this talk at least, I'll be focusing more on the qualitative sides. So a doctor is seeing a patient, the patient describes his list of symptoms, and then you want to look at the database of all the patients that you've ever seen so far. How do you efficiently retrieve which patient's symptoms are most similar to what you're seeing right now? Similarly, for each disease, you might say that this disease typically causes these symptoms. How do you efficiently retrieve which disease is most likely to be the cause of this particular patient, and so on? Okay. So to explain some of the concepts, I'll do a brief detour through books. So let us say that let's say that you have a book and you have a library of books and you're trying to put metadata on all of these books so that you can efficiently retrieve the book that you want or books that you like. So you could have all kinds of metadata. Standard metadata, you'll have author, title, some description, and some additional attributes which are not really useful from a retrieval perspective. And then you might want to search for things saying, I'm interested in funny crime fiction books set in Bangalore. If you were to type in this text string and you were to try to search all of these descriptions, you may not get anything at all because these exact phrases may not be present in the description. So this is really what happens most of the time. If you were to try Google, you were to put in this thing, unless these exact words match in the description of some book, you're not going to get that book back as a hit. So the next logical thing is how people attach tags to items, and people can put whatever tags or keywords they want with items. So you could say that the book is a collection of keywords or tags, and this is what we have for journal articles also, we put keywords. So you could say that this book has these following keywords: spy, thriller, humorous, Chennai. And then if you were to search on related tags, you might get it. But again here the tag to tag matching is an exact spelling match. So if you're searching for crime fiction, even though spy, thriller are similar notions, it's not going to find that book because the concept crime fiction is not really related, the tag crime fiction is not really related to the tag thriller by any structure so far. So you may get this book or you may not get this book. Okay. So next, so the next logical thing to do is to organize the tags in a hierarchy. So you could say that let me try to define the universe of all books. Let me try to organize all of these tags in some particular structure. So you could say that a book is either a fictional, is either of type fiction or of non-fiction. If it's of fiction, it could be a pictorial, it could be mystery. Mystery can further be divided into crime fiction and spy fiction, and so on. So once you define all of these classes, and more importantly the relationship between these classes, that is in a very simplistic term an ontology. So an ontology is a structured arrangement of classes along with the relationship between those classes. Okay. So now, given this particular ontology, you could say that these two terms are very close to each other in the ontology, so they are in some sense similar to each other, whereas this term and autobiography are very far from each other in the ontology, so they might be very distinctive. So let us say that we have 4 books out here, 5 books out here. There is this red one which has been tagged with superhero, that one with anime, spy, self-help and biography. So suppose the library has this particular system, and you go to the library and say that you want a mystery book. There is no book explicitly tagged with mystery, but it knows, because of this ontology, that crime fiction books are also a type of mystery books. It knows that spy fiction books are also a type of mystery books. So the library system could retrieve that spy book and give it to you. Similarly, if you go and say that you want a comic, and using the same logic, if it were to give you this red book, and you say that I've already read the red book, then it can say that the most similar book for this is the anime book. It could suggest that for you. Okay. So this is just to build up an intuition about what does similarity mean. In general, things which are close to each other in the ontology are similar to each other by some measure. But we also need to take into account the information content. Let us say that we have a library which is dealing only with fiction books. Let's say it has 5 books tagged explicitly with the word fiction, 10 of them tagged with mystery, 15 with crime, and so on. Now, if you were to say, if somebody were to come in and say, I want a fiction book. Now, this particular term, fiction, no longer has any information content at all, because every single book in the library is a fiction book. Okay. Similarly, if you were to say I want a spy book, now that one has heavy information content, because the overall percentage of books which are spy is very small. So in some sense, the probability of a book in the library being associated with a class determines the information content of that particular class. Okay. At the same time, we know that superhero comics are also a type of comics. So what we can do is say that if a book is classified as superhero, implicitly it's also classified as a comic, it's also classified as a pictorial book, it's also implicitly classified as a fiction book. So if you were to spread that information up the tree, you'd really have that picture out there. Okay. So there are 100 books overall, all of which are a fiction, 30 of them are mystery, 15 crime, 5 spy, and so on. Based on all those numbers, you can assign an information content value to every node in this ontology. So information content, you can take the probability and do a minus log of that. So if you were to compute the information content for fiction, that is probability 1, because there are 100 books overall. There are 100 books marked with fiction, so the probability is 1, so the information content is 0. Okay. Whereas if you were to take spy, then the probability for that would be 5 out of 100. If you take a log of that, negative log of that, it would be fairly high. So I can say that this notion of information content captures to some extent which terms are important or not within that particular library system. And immediately after that, you have this notion of similarity. So you're trying to see how similar are two terms in this ontology. So for instance, how similar are superhero comics and anime comics? And we had the intuition in the previous slide that two things are similar if they are close to each other in the ontology. By being close together in the ontology, you mean that when you do the least common ancestor, you reach a node very quickly. So one way of quantifying that is to say that this one, one at the bottom. Okay. Oops, not that.
You have to pull the trigger.
Okay. Pull the trigger. This. Okay. So if you want to quantify the similarity between two nodes, what you can do is find the least common ancestor, and the information content of the least common ancestor is a measure of the similarity between those two nodes. So if you take two nodes which are very far away from each other, like superhero and spy, the least common ancestor would end up being close to the root, and the root would have a large number associated with it and a very small information content. If two things are very close to each other in the ontology, you'll reach their least common ancestor very quickly, and it will most probably have a better information content. Okay. So all of this you could use for a library retrieval system, where you can think of the first term that you're putting in as your query term, and the second term is a term associated with the books. So you're putting in one term out here, N1. So you're saying that I'm interested in a spy book and so on. And it can go and query every single node and say what is the similarity of your term with every other term in this ontology, and say which of them are similar to what you want or not. Okay. So really what we have is two dimensions. You have an ontology which is telling you what is the structure of knowledge in the book universe, what are books about. Books are, what are the categories of books, what are the subclasses of books, and so on. So this is a pure ontology. And the second, you have a domain, which might be a library of books or any other domain, and you're mapping elements of this domain to terms in this ontology. And an element in this domain could be mapped to multiple terms in the ontology. You could have a superhero book with the setting being Chennai. You could have an anime book, which is an international book, and so on. So every element in this particular domain is being associated with a set of nodes in that ontology, and that's what defines that book. Okay. Then the question is, so now let's revisit the original problem. You want to search for certain books, and that is being specified as certain terms in the ontology. You're interested in crime books set in Chennai, which have some humor in them. Those are all different terms in the ontology. So you're specifying a collection of terms. Each one of the books in the library is also associated with a set of terms in the ontology. Right? Every book is also marked with something. So what you're doing is taking your set of query terms, you're comparing it with the terms associated with each book, computing the similarity. And whichever book is the most similar to your query terms, that is what comes out as a result. So, so there's some very simple metrics out here which define how do you compute the similarity. This is still an ongoing area of research. This measure is called the Resnick measure, and this is what seems to work well in practice.
So how would you contrast this with Google search?
Google search does not have any semantics or ontology behind it, right? It's pretty much a plain text search. So it's mainly a syntactic search. This is more of a semantic.
Database you have, this is not the mark or only social picture with related terms. That is, I feel, it's based. I had a similar question. So he has this Wikipedia-based ontology, something like a Wikipedia,
Yeah.
on which he's kind of displaying Mahatma maybe related to Nehru and all.
Those are usually based on common terms shared between those two pages. So what you would also do is
It's like your ontology, somebody has marked it.
yeah.
And even the book, somebody has marked it.
Yeah, so first you could do related objects even without ontologies. For instance, you could just look at individual words or phrases and then look for each phrase. Is it present across all articles? What is the information content of each phrase? Does it disproportionately occur in some articles versus others, or is it uniformly distributed across articles? Based on those, you identify enriched thing, and those kind of become keywords for your article. And two articles which have similar set of keyword things could be related to each other. So you could do a lot of these things without any real ontology behind the scenes. But if you want to have that fine distinguishing power, then it makes sense to create an ontology.
What I'm going to ask is, like, right now the ontologies cannot be automatically generated from the document. It's kind of handmade.
Yeah.
Somebody sits and says, okay, this is how I'm starting file.
Yes.
Well, I mean, I think nobody has done any kind of an automatic ontology generation from a book or something.
The one set of people who did automatic ontologies was the Wolfram Alpha people. They had created some system. There was a Wolfram Alpha search engine, right? A couple of years ago, it made a lot of news. So that time they had some process where they would do text mining and automatically pick out which words were related to a particular domain and create a very, very lightweight taxonomy out of it. I'm not sure how they did it because they didn't publish it, but I saw the video and it was quite nice the way they did it. Okay. So some of the challenges are that creating an ontology is very, very difficult. And the same domain, if you want to create an ontology for books, no two experts on books will agree on the correctness of that ontology, because there is no correct thing. You could say that I'll start dividing my books into large books, heavy books and small books, and you're not wrong. I mean, that is one dimension that's important for you. You could divide it that way. Some people would say, no, I want to go it based on genre. I'll do crime and this at the first level. Some will say, no, I'll do it based on language. That's also an important dimension. Some will say, I'll do it on author's first name. You really can't. So it all depends on what do you want to use your ontology for. So the way you create your ontology should strictly be defined by what is your end goal in mind. If you simply say that I'm trying to structure all the knowledge about this domain, it will never work. Okay. So in biology is completely driven by ontologies. There is a portal called the NCBO Ontology Portal, National Center for Biomedical Ontologies. There are around 350 ontologies out there. Some of them have 40,000 terms arranged in some very complex structure, and most of those ontologies have ontology maintainers. They start off as a research project, they publish their ontology, they get a paper out of it, then they're stuck with it for life, because every single expert will start giving them suggestions saying, but why is this term here, should it not be out there, can you also add this thing? And once they add 3 or 4 things into one part of the ontology, it looks terrible, so they have to redo their ontology structure. So it becomes pretty much a lifetime task maintaining an ontology. And every time you adjust your ontology, whatever elements you mapped onto that ontology, so you have your entire library of books, and each one of those books you mapped it onto this ontology. If you change your structure, you need to go and remap them to that thing. Okay. And if you remember, recently there was a lot of buzz about Wikipedia. Wikipedia was creating all of these authors. Wikipedia also has a very, very lightweight ontology, more like categories is what they call it. So they were trying to redo their authors categories. So they created a new category called women authors, and they started moving the authors from the women writers into the women authors category. So there's a lot of protest saying we don't want to be labeled as women authors, we just want to be labeled as authors who are also women. Okay. So then it becomes about how do you deal with the cross products of ontologies. So women authors, is it actually a cross product to ontologies? There's an author, there's a gender, you're doing a cross product of all those and creating these out here, or things like that. So these can be quite controversial. Okay. So now coming back to biology, this is a real example from a published paper. A doctor, when they carry out a study, they're going to write up their notes in something like this. A lot of the next-gen sequencing buzz is because of its ability to diagnose rare disorders. So a patient walks in and the doctor has absolutely no clue what's going on. They do a lot of studies, they do a lot of measurements, they find out a lot of symptoms, but they're not able to narrow down what is the cause. That is when right now, these days at least, they resort to next-gen sequencing. They sequence their entire genome, they look for the mutations, try to find out which gene might be the cause. So this is one particular case where they had twins, 14-year-old twins, and they found a lot of issues. I'll highlight some of them. They said that they had symptoms of dystonia, there was something in a hypercoagulable state, there was hypotonia, developmental decay, leukomalacia. So we don't have to understand all of these terms, but these are all different observations that the doctor made about those twins. There was rigidity, there was tremor, and so on, seizures, etc. So we have all of these different terms which we'll call phenotypes. Phenotype refers to something which you can observe in a human being, either in the form, that is, you can observe that the forehead is very compressed or very large, the nose is very depressed. So it's either in the form or it's in the function, saying the heart is beating at a much higher rate than expected, the breathing is very shallow. Those are all function level things. Or in the behavior, saying there are uncontrollable tremors and seizures and so on. Any one of those things that you can observe is called a phenotype. So you have this whole collection of phenotypes that you're observing in these twins, but you don't know what is the cause of those. So then what the doctor wants is a database where all the diseases have also been marked with phenotypes. Let us say for every disease that is out there, the clinicians had written saying, if you have this disease, these are the phenotypes that typically show up. If you have a mutation in this particular gene, these are the things that will show up. So if genes were cross-referenced with phenotypes, if diseases were cross-referenced with phenotypes, then once a patient walks in with this collection, we take these phenotypes and say which gene is most similar to this, which disease is most similar to this collection of phenotypes, and based on that you narrow down your diagnosis. Okay. But for this, and these are other pediatric cases. There was a patient, there was a male child born at 13 months, and at 13 months they had all of these issues. There was someone else born and so on. Okay, with copper levels were low in the blood and so on, things like that. These are all cases published in the last couple of years. Okay. So what we need is a comprehensive controlled vocabulary or an ontology for phenotypes. We need a way of taking all of this doctor's report, automatically extracting all of these phenotypes using text mining and mapping to terms in this ontology. And based on that, we need to identify the disease and genes associated with them. So already defined what a phenotype is. So around 4 years ago, a group of researchers sat and created this ontology, a group of German researchers. So around 9,500 different abnormalities that they organized into a huge acyclic graph. So for instance, this is something for maternal diabetes. They give a description of the term, and they say that it's a type of diabetes. It's also a type of prenatal maternal abnormality. And they organize all of this into this huge structure. It's no longer just a simple tree structure, it's actually a directed acyclic graph, because you can see that there are things like maternal diabetes. It is both a diabetes and it is also a maternal abnormality. So a node can have multiple parents in it.
So this terminology is across the world, or is it US?
That's a good point. So you'll never see just one ontology for anything. You'll always have multiple competing ontologies. So what they did was they went through a resource called OMIM, Online Mendelian Inheritance in Man. There's all the Mendelian disorders are typically described on that website. So they went through all of those pages. They tried to figure out what all are the terms that people are using in real case studies, and they tried to merge two terms which are very similar to each other and so on, and created this. It will work. These abnormalities are common across all populations. They are nothing specific to a particular population. Okay. So they'll have things like, abnormality of the face. They'll say abnormality of the lips, and too large, too small. All of those terms are fairly general. So, and
doctors, for example, use the ICD codes, ICD-9, and now ICD-10.
Yes.
And these are all the codes of disease. So it's just that now you're overlaying a structure.
Yes, on top of this. So for instance, we were doing a couple of case studies in here with Center for Human Genetics. We took the exact case report given by the doctor out there, and all of the terms that were present in it we could find in this ontology. So that gives us some confidence. It's fairly general. At the same time, they are also maintaining it, so people keep writing and they keep updating that ontology on a weekly basis. This is open. It's open in the sense that if you want to check for phenotypes, it will be there. It doesn't give a diagnosis. It's just a phenotype ontology. Okay, just like how if you have that ontology for books, it is telling you that there is something called crime fiction, that it's a part of fiction and all, but it's not giving you a book in itself. It's just about books, how do you organize. Similarly, these are all the different phenotypes, but it's not going to tell you which disease is associated with it. It's not going to find an individual case. That will come from the mapping of diseases. Just the way we said there's a library, and individual books in the library are mapped onto this ontology. So similarly, there's a disease collection, and individual diseases will be mapped onto this phenotype ontology. and there will be individual patients, and the patients will again be mapped onto this ontology. Okay? So the ontology is serving as a common framework for all of those other objects to be mapped onto it. Okay? And there are tools dedicated for displaying ontologies, for creating ontologies and all. A very popular one is called OBO Edit, and you can see that these are all the different main physiological systems in the body, and behind that is that whole graph sitting behind it. So then what they did was they went to this resource called OMIM, where all of the Mendelian disorders are described. This is one called DRD, dopa responsive dystonia. Each one of these disorders has a huge amount of clinical text written about it. So all of the doctors around the world who find an interesting case study will either publish a report, or they will go and submit some data here and so on. So for instance, this was a 1976 article where they described something, there's a 1986 article where they described something, and so on. So what they did was they went through each one of these approximately 3,700 Mendelian disorders. They go through the page, they try to spot all the phenotypes that are mentioned out here and say that these are the phenotypes associated with this disease. Okay? So just the way we're saying the library, all the books have to be mapped onto this ontology, so that's what they've done. They created the ontology also, and they went through this resource mapping the diseases to the ontology.
People mind these texts of this and you have these keywords coming on top, right? Which are they talking of the most? Have they done for many people?
They've done a lot of that. We have done text mining engines of our own. Here also it was partly automated. They ran a text mining thing to pull out common terms. And then, but the mapping was done manually. So the text mining was used as a preprocessor to suggest things which were then mapped manually. So based on all that, after going through the DRD page in great detail, they said these are all the different ontology terms which they mapped onto DRD. Okay? So like for every single disease, they'll identify 10 or 12 terms, phenotype terms, which they'll map to the disease. So now what do we have? We have this human phenotype ontology which is serving as a common structure. We have all of these 3,700 inherited diseases. Each one of those diseases has been associated with a set of phenotype terms. Okay? Ranging anywhere from 2 to 12 phenotype terms are associated. Now what we want is we want a query matcher. We have a patient who's walked in. The patient has a set of phenotypes. We just want to put all of these phenotypes into some query search engine and ask it to compare these phenotypes to every one of those diseases. And whichever diseases phenotypes most resemble this, we want that ranked disease list. Okay? So this is in some sense a diagnosis engine then. Okay? And the similarity metric is done exactly the way I described before. You have this collection of phenotypes, you're comparing it with some other collection of phenotypes, and we use the Resnick similarity measure.
Is the Euclidean distance which you take between the feature vectors?
No, because you have to take the tree distance and that information content into account, right? So because of that you can't do simple Euclidean.
Yeah, you can treat the tree distance itself as a feature, in which case it's linearized in the other different dimension, different transformation space.
Yeah, it doesn't work as well if you just work on the tree distance, if you ignore the information content, because some phenotypes will be so common that they shouldn't contribute as much as phenotypes which are rare.
Yeah, I'm just saying you can linearize, you can treat it as two components in the tree.
Yeah, these are all areas of work. There are 5 different metrics which they compared, and this one seemed to work well in this particular domain, but it's still an active area of research.
So is there reasonable certainty or reasonable evidence that really these diseases are separable, at least using convex separations in the feature space chosen, or could you come up with a whole, something like I can have a set of parameters, I can have a collection of parameters,
Yes.
all of which represents one disease, but right in the centroid of that region, it will not represent that disease.
Yeah, people haven't looked at it from a mathematical perspective like that. They're simply saying empirically, because you also want to work with a set of features which are likely to occur together. It is always possible that you choose a collection of phenotypes which will sort of completely break this algorithm also, right? But
So all I'm saying is, is it convex? Is the class a convex body in the space dimensions chosen, in the feature dimensions chosen? I mean, I suppose people would believe that it's convex, but have you looked to see to what extent you have non-convex?
no, these are all things which came out only in the last couple of months. So really, that kind of mathematical work hasn't happened yet.
Because if it's not convex and it has holes in it, it will become, you need to add dimensions before you can actually separate the classes.
Yeah, most of the time people look on it only for a ranking thing, and as long as it shows up in the top 20 or 30 across all of the genes. So another reason why it's not important to get it absolutely right is that you're also looking amongst all those 30,000, 50,000 mutations. Only a handful of them are actually dangerous. Each one of the mutations, purely through bioinformatics, you can identify is it likely to cause the change in the protein or not. And you'll end up with around 50 or 60 of those which are likely to be harmful. Now, among the list of genes which shows up out here, the intersection of this set of genes with the genes which have mutations is usually only 2 or 3. So in which case you're pretty much done. So nobody has really done the theoretical work for these areas yet, because in practice they seem to work well. Okay. So here, for instance, if you were to take that same neurological case and take all of these keywords, these are all the set of keywords that are mentioned out there, the phenotypes that are mentioned out here. And if you were to run it through the Tresmix similarity measure, it would identify the correct disease, this dopa responsive dystonia, right at the top. And what is nice is that there's almost no exact syntactic match between the actual phenotypes associated with the disease and the phenotypes that you gave in as the individual's phenotypes. So these were all the phenotypes that the clinician mentioned for this particular case. These are all the phenotypes that are actually associated with the disease, and if you were to look at it, there's almost no overlap. But still it came up as a top ranking one. And the reason it came up as a top ranking one is because that things are close to each other in the ontology. So this is one of the terms that was associated with the disease, and this was one of the terms that the physician used for this particular case, and they're sort of close to each other because of, and it's a collection of all of those together which brought it as a top ranking one. So basically in the real world, the things that it needs to handle is imprecision. When you map a disease to that phenotype ontology, it's being done by an expert. So they looked at multiple such disease cases and very carefully mapped the disease to that ontology. They choose very, very specific terms which are usually further deeper down in the ontology. But when a clinician is studying somebody and they're creating that case report, they haven't spent as much time. So they'll see phenotypes which are much higher up in the ontology, which are shallow phenotypes. So it needs to be able to handle imprecision, where what you're querying may be an ancestor of what is actually associated with the disease. You may observe only a subset of the phenotypes which are actually there. In the first examination, you may notice only 2 or 3 of those phenotypes. So it needs to handle incompleteness, and sometimes it might be a mixture of 2 or 3 things which are going on in the individual. You may say that he has these phenotypes, he also has high temperature. The high temperature may be completely unrelated to it. So it needs to handle a bit of noise. So what they have done is they've done some simulations and taken around 4 or 5 of these diseases, and they've tried to simulate imprecision, incompleteness, noise, and shown that this particular metric seems to work well in practice. But beyond that, no theoretical proofs have been done here. Okay. And now all of this is based on whatever knowledge you have about human diseases. So somebody has seen a human family with a disease, they noted down the phenotypes and they create the mapping. So what we are doing is we are taking the patient symptoms, we are mapping it onto the human phenotype ontology, based on that prioritizing the genes. But a lot more work has actually been done in the animal domain. So in the mouse, for instance, every single mouse gene, they have tried to either mutate it or knock out the gene and see what happens to the mouse. So every mouse gene is being associated with a set of mouse phenotypes. And they're doing this in the fruit flies, they're doing it in yeast and so on. So in these other organisms, each gene is associated with a set of phenotypes specific to that organism. So if this particular approach doesn't work, what people are doing this year is that you take the patient symptoms, you map it to the human phenotype ontology, then you create a mapper between the human phenotype ontology and the mammalian phenotype ontology, which is used to describe mice phenotypes. So the mice phenotype work started 10-20 years ago when they systematically started knocking off individual mouse genes. So this ontology and this ontology are completely different from each other. So now you take the symptoms, you map it here. There's a bridging ontology called Pato. You use that to map it here. Then you say that, okay, these are the mouse phenotypes I'm looking for. You see what are the genes responsible for those mouse phenotypes. Then you look up human mouse homology models which say that this human gene is the same as that particular mouse gene, and you bring it back to the human gene. So they've shown 1 or 2 case studies where this entire approach seems to work. But as you can see, there are lots of places where it can go wrong. Mapping whether you actually captured the human phenotype accurately or not, and mapping from one ontology to another is very, very complicated. It depends on the completeness of that, and all of this obviously assumes that mouse genes and human genes are sort of similar to each other. So that underlying assumption is also there. Okay. So all of this is a very active area of research in text mining, semantics, and logic. Not many people have looked at it from a quantitative perspective to see which of the metrics and what are the properties of these metrics. That work has not yet happened. Okay. So yeah, so the autism candidate genes they found out via mouse phenomics. Okay. And yeah, that's all I have. Okay.
Thank you, Dr. Vamsi, for such an insightful talk on ontologies and how we could potentially use it for automating the disease diagnosis process. We would like to call upon Professor Raghavan to hand over the conversation.